Good afternoon, dear students. Now we are going to discuss today about gene mutation. Now, what is a mutation? Any sudden heritable change in the gene leads to mutation. Okay, a sudden heritable qualitative or quantitative change in the genetic makeup of an organism leads to mutation. Now, what is a gene? We have studied what is a gene. It is a segment of DNA, or you can say it is a sequence of nucleotide. It is a polynucleotide sequence. So any qualitative or quantitative change in the genetic makeup of an organism in the gene leads to mutation. Okay, and the term mutation was first coined by Hugo de Varis to describe the sudden heritable phenotypic change, okay, in the flower primrose. Okay, and then further other scientists like T.H. Morgan and H.J. Muller, okay, they studied about various mutations taking place and how mutations can be brought about. But mutation, the term mutation was first used by Hugo de Varis. Now, see, we have studied units of a gene. So, the gene as a unit of mutation, that unit of a gene where mutation take place is called as a muton. So, the unit of gene where mutation take place is called as a muton. Now, there are certain regions in a gene where mutations occur more frequently. Such spots are known as hot spots. Okay. So, what is a mutation? Any sudden heritable change occurring in the DNA, qualitative or quantitative change occurring in the DNA is called as mutation. And the term was first coined by Hugo de Varis. Mutation may be harmful, beneficial or neutral. Okay. Now, one more thing I would like to tell you. What is the rate of mutation? See, the frequency of spontaneous mutation, what I'm saying, the frequency of spontaneous mutation is very low, that is 10 raised to minus 7 to 10 raised to minus 12 per organism. Okay, so the frequency of occurrence of spontaneous mutation in any organism is very low, 10 raised to minus 7 to 10 raised to minus 12, and the rate of detectable mutation in a gene is also low, that is 1 in 10 raised to 6. Okay, so the rate of mutation is extremely low. One mutation out of 10 raised to 9 nucleotides per generation, or we can say per gene, like 1 in 10 raised to 6, or per organism. It's very low, frequency is very low, 10 raised to minus 7 to 10 raised to minus 9. Now, if we look at whether mutations are random or whether they are directed towards any objective, then we come to know that mutations are random. They do not occur in an organism because the organism wants the change to occur. Mutations occur randomly. That means mutation occur in a Darwinism manner and not in Lamarckian manner. Okay, that means that if mutations do not occur in an organism because the organism wants the change to take place in response to the environmental condition. For example, according to Lamarck's theory, changes or evolutionary changes take place in an organism because the organism wants the change on its part, okay, according to the changing environmental conditions. But mutations are not like that. Mutations are random. According to neo-Darwinism also, mutations are random. They abruptly occur in an organism. And if you look at a unicellular organism and a multicellular organism, then unicellular organisms are single celled organism, and the germ cell as well as the vegetative cell are the same. Okay. So, because somatic cells and germ cells is the same cell, unicellular organisms are more subjected to mutation. They are more prone to undergo mutation as compared to multicellular organism, because in case of multicellular organism, the germ cells and somatic cells are different. The germ cells or reproductive cells are much more protected, right? So, unicellular organisms are more exposed or more prone to undergo mutation as compared to multicellular organism. Now, mutations are variously classified. There are different types of mutation. Now, depending upon what is the effect of mutation on the phenotype of an organism, external features of an organism, depending upon the what are the effects of mutation on the phenotype of an organism, mutations are classified as lethal mutations, subvital mutation and supervital mutation. What are lethal mutation? It leads to the death of the organism or cell in which it takes place. Okay. Lethal mutations are those which result in the death of the cell or organism in which it takes place. Subvital mutations are those in which the chances of survival of the organism become less, and supervital mutations are those mutations, okay, in which you can say there is improvement in the biological fitness of the organism under certain conditions. Okay, supervital mutations are those in which there is improvement in the biological fitness of the organism, okay, according to certain conditions. So depending upon the effect of phenotype, mutations may be lethal, subvital or supervital. Now we are here going to study molecular basis of gene mutation. If you look at molecular basis of gene mutation, right, there are point mutations. Okay, point mutation means what? If I say mutation has taken place in an organism, it may not be a long segment of DNA where changes have been occurring. Okay, the mutation or changes in a long segment of mutation, changes in the long segment of a nucleotide are not needed for mutation to take place. Mutation can take place even if there is change in a single nucleotide base. Okay, mutation can occur also when there is change in a single nucleotide base. Okay, so mutations may be point mutation. What is a point mutation? Where there is substitution of one nucleotide by another at one point, or there is deletion of one base, insertion of one base. Okay, so point mutation means what? There is change in a single nucleotide, maybe substitution. Okay, mutations can also be of insertion type of mutation or deletion type of mutation. Insertion means what? One base is added extra, one or more bases are inserted. Deletion means what? One or more bases are deleted from the nucleotide sequence, right? So a point mutation where there is change in a single nucleotide base, substitution of one base by another base leads to silent mutation or missense mutation. We call them base pair substitution. Point mutations are of two types again, or point mutations can be variously classified as base pair substitution because one base is substituted by another base. Base pair substitution leads to various type of mutations. They are missense mutation, nonsense mutation or silent mutation, and mutations in termination codon that we are going to study later. Okay, so base pair substitution where one base is substituted by another base leads to missense mutation or nonsense mutation or silent mutation or mutation in termination codon. Okay, when mutation occur because of deletion or addition of a base, it is called as frame shift mutation. Okay, so frame shift mutations are those wherein there is either addition of one or more base or deletion of one or more base which leads to shift in the reading frame. Okay, so addition or deletion of base leads to frame shift mutation, and substitution of one base by another leads to missense or nonsense or silent mutations. Okay, so let us study now various type of mutations. What is a point mutation? A point mutation, gene mutation which change the DNA sequence. A point mutation is a change in a single base pair. Okay, there is change in a single base pair. It involves a base substitution. Okay, point mutation involves base substitution. We also call it base pair substitution or switches. Now, base substitution means what? Suppose we have a DNA sequence that is shown here, like it starts from 5' end to 3' end. A, C, G, C, T, A, G, A, T, C. Okay. Now, suppose there is change in one base. Say for example, A is substituted by say C. Okay. So what will happen if A is substituted by T? Then AT pairing will, if A is substituted by C, what will happen? AT pairing will change to CG pairing. Okay. So base pair substitution may bring about conversion of AT pairing into CG pairing or vice versa, like that. Okay. Now a transition. Now base pair substitution are of two types. Base pair substitutions are of two types. Number one, transition, and number two, transversion. Okay. Base pair substitutions are of two types. Transition, number two, transversion. What is a transition type of mutation? A transition type of mutation is a change wherein one purine is substituted by another purine, or one pyrimidine is substituted by another pyrimidine. Okay. What are the two purine bases? Adenine and guanine. Okay. And what are the pyrimidine bases in a DNA? Cytosine and thymine. So when I say transition type of mutation has taken place means what? One purine is substituted by another purine. That means A is substituted by G. That means originally there was A in the chain. Now it was substituted by G. So if the original base in the chain was A, then there was AT pairing. Now if the base A is substituted by G, then AT pairing will change to GC pairing, because A is substituted by G, then in the new strand G against G, a complementary base C will be added. So in the nucleotide sequence there will be change in one base pair, that is originally it was AT, now it changes to GC. Okay. So like that changes will take place. Say for example, if we look here, this is a base A. Okay, so against the base A, suppose this is a nucleotide sequence and there is base A. So against the base A there will be T. That means in the nucleotide sequence there will be AT pairing. Now if A in the nucleotide sequence undergoes, or if there occur transition type of mutation, then A will be substituted by G. A will be substituted by G. So A is substituted by G. Now when replication will take place, against the G which base will be added? C. Against G which base will be added? C. So originally there was AT pairing. Because of transition type of mutation, A was changed to C, and hence now there will be GC or CG pairing. So in the nucleotide sequence, originally there was AT pairing, now it has changed to GC pairing. Such a kind of mutation is called as a base pair substitution. It is a type of point mutation because mutation has taken place only in one of the base, and this type of mutation is called as transition mutation. Okay, where one purine is substituted by another purine, or one pyrimidine is substituted by another pyrimidine. Like adenine will be substituted by guanine, or guanine will be substituted by adenine. Similarly, there are two pyrimidine bases, cytosine and thymine. So cytosine will be replaced by thymine, or thymine may be replaced by cytosine. Now, what is a transversion type of mutation? Transversion type of mutation is the one in which purine is substituted by pyrimidine, or pyrimidine is substituted by purine. Like say adenine is substituted by thymine, right? So then there will be ATTA only. But if adenine is substituted by cytosine, of adenine is substituted by cytosine. Adenine is a purine base and cytosine is a pyrimidine base. So if adenine is substituted by cytosine, then AT pairing will change to CG pairing, as I told you now. Okay. So these are the two different type of base pair substitution, like transition or transversion. But out of the two, transition mutations are more common than transversion. Okay, so I hope you understood what is a transition type of mutation and what is a transversion type of mutation. So if you look at the example of a point mutation, change in one of the nucleotide. For example, we've just written a statement: the fat cat ate the rat. Now if there is change in single base. Okay, the fat cat. Say for example, the C was replaced by H, right? C was replaced by H. So the word cat changed into hat. The fat hat ate the rat. So if there is change in one base of a nucleotide, it will change the codon. Okay, it will change the entire codon in the mRNA, and hence the anticodon in the tRNA will be different, and hence the amino acid that will be added during protein synthesis will be different, and change in a single base pair will lead to change in one codon, leading to change in one anticodon, leading to change in one amino acid in the protein. If there is a change in only one amino acid of the protein, the whole protein may become totally inactive or partially inactive. It may be partially active or it may become totally non-functional. So even there is a change in a single base pair, substitution of a single base pair by another base pair, it can have a drastic effect on the phenotype of an organism. Okay, so that is about point mutation. Now they are of two types. Point mutations on the basis of molecular basis of mutation, there are two types of mutation. Number one, base pair substitution, or also called as switches, and number two, frame shift mutation. Okay, so base pair mutation or frame shift mutation. Base pair substitutions are of two types. Number one, transition, and number two, transversion. Now base pair substitutions are the simplest type of mutation take place. What does it involve? It involves alteration in one single nucleotide base pair. There is a change in a single base pair of an organism. Okay, so a base substitution usually leads to base pair substitution, of course. Of course. So if there is base substitution in the template strand of the DNA, say for example, just as I told you, if there was say adenine in the template strand, and if that adenine was substituted by say guanine in the template strand, then in the next generation the AT pairing changes to CG pairing, GC pairing. Okay, so a base pair substitution, a base substitution usually leads to base pair substitution and change in the nucleotide sequence. Base pair substitutions are of two types, as I told you, transition and transversion. What is a transition? Purine is replaced by purine. One purine replaced by another purine. Like A can be replaced by G or G by A. Or a pyrimidine is replaced by a pyrimidine. Like C is replaced by T or T is replaced by C. Okay. And what is a transversion type of mutation? Wherein a purine is replaced by a pyrimidine or a pyrimidine is replaced by a purine. Now, if I say purine is replaced by pyrimidine, that means what? A can be replaced by C or A can be replaced by T. Right? So, one base can undergo only one type of transition, but one base can undergo two types of transversion. Okay, so A can be replaced by C or T, G can be replaced by C or T. If I say a pyrimidine is replaced by purine, then C can be replaced by G or A and T can be replaced by G or A. The possible number of transition are, usually transition type of mutations occur more as compared to transversions. Okay, so the number of possible transversion is twice the number of possible transitions, but transition mutations are more frequent in vivo as compared to transversion. Now, on the basis of effect of phenotype on mutation or molecular basis of mutation, most common phenotype in natural population of the organism is called as wild type phenotype. Okay, whatever occurs naturally, okay, when microorganisms occur, the naturally occurring microorganisms are called as wild type phenotype. Okay, so the most common phenotype in natural population of the organism is called as wild type phenotype. Now, mutation refers to change in the wild type phenotype. Okay, mutation refers to changes that are brought out in the wild type phenotype. So any change in the wild type phenotype at molecular level, at genetic level, leads to mutation. Mutation may be forward, mutation may be reverse type of mutation. We'll come to now presently we are studying forward mutation. Okay, then we will study about reverse mutations. Now, A mutation that alters the wild type phenotype is called as forward mutation. Now, what is a reverse mutation? Reversion mutation. That also we are going to study. A mutation which will change a mutant phenotype back into the wild type. Reversion. So now let us study about forward mutation, and the first one, that is base pair substitution. Base pair substitution are type of point mutation. What a point mutation? Where change occur at only one point in a single nucleotide base. Okay, at a single point where one or the other bases is replaced by other base. So base pair substitutions are either transition type or transversion type. Base pair substitution, also called as switches, are those mutations in which there is change in a single nucleotide leading to change in base pair. Okay, base, that is why the name base pair substitution, where one base is replaced or substituted by another base. Base pair substitutions bring about four type of mutations. Number one, missense mutation. Number two, nonsense mutation. Number three, silent or neutral mutation. And number four, mutation in termination codon. What is a missense mutation? Missense mutation is the one in which a base is substituted that alters the codon in the mRNA, resulting in a different amino acid in the protein chain. Okay, now let us understand what is a missense mutation. Missense mutations are those in which there is change in a single amino acid in a polypeptide chain. Okay, the missense mutation result as they take place as a result of change in a single amino acid in the protein chain. Why there is a change in the single amino acid as compared to the normal one? Because if there is change in a single nucleotide base pair, right? If there is a change in a single nucleotide base pair, if there is single base pair substitution, but one base pair is substituted by another base pair, then this will lead to change in the codon of the mRNA. If change take place in the gene, okay, say if adenine is substituted by guanine, for example, or if C is substituted by T as shown in this example, this is TCA. Because of mutation, it has changed to TTA. That means C, the central one, C is substituted by T. If one base is substituted by another base, then the complementary base in the newly synthesized DNA strand will also be different. That means TCA pairs with what? AGT. So now the C was changed to T. So instead of CG pairing, now there is TA pairing. Okay. So TTA changes to, AGT changes to AAT. So change in a single nucleotide base pair leads to alteration in the codon of the mRNA, and hence it also leads to change in the anticodon. Okay, so it leads to change in the codon of the mRNA, and hence a different amino acid is added. A different amino acid is added during protein synthesis. So the first one here is showing a normal codon UCA, which codes for, say, serine. So there is serine in the polypeptide chain. Now if there is mutation, the codon of the mRNA also changes and it adds leucine instead of serine. So what happens here? In the polypeptide chain, in the normal chain there was serine amino acid, and because of base pair substitution, it has changed to leucine. Okay, serine is replaced by leucine. So replacement of one amino acid by another, substitution of one amino acid by another amino acid leads to missense mutation. So missense mutations are those mutations in which one amino acid is replaced or substituted by another amino acid. The changed protein sequence may be totally non-functional. It may have drastic effect on the phenotype of the organism, or the protein may be partially functional. Why amino acid changes? As I told you now, if there is change in a single codon, it will lead to change in the amino acid. Why there is a change in the codon? Because there is base pair substitution. Okay, so if there substitution of base takes place in the gene, it alters the codon in the mRNA, leading to addition of a different amino acid, and this causes missense mutation. The best example of missense mutation in human beings is the sickle cell anemia. Okay, sickle cell disease is the result of one nucleotide substitution. You know, it's a genetic problem. We say sickle cell anemia is a hereditary transmitted from one generation to another generation. Okay, and sickle cell anemia is a genetic disease. What happens in sickle cell anemia? There is change in only one base. Okay, so substitution of one base by another base in the gene leads to change in the codon in the mRNA, leading to addition of different amino acid during protein synthesis. There are, see, the two hemoglobin genes, two alpha and two beta genes. So in the beta globin gene, if you look at from N-terminal end, from the N-terminal end of the beta globin gene, there is, if you look at the protein chain of hemoglobin, then from the N-terminal end, the sixth amino acid is glutamic acid. And if this sixth amino acid, glutamic acid, is replaced by valine, it leads to sickle cell anemia. Such a drastic change occur because of replacement or substitution of one amino acid by another. Such kind of mutations are called as missense mutation. Base pair substitution that result in substitution of one amino acid by another is called as missense mutation. Missense mutation substitute one amino acid for another. Missense mutation have very large and drastic effect, while others have very minimal effect. It depends on where the mutation is taking place. Okay, where the mutation occurs in the protein structure and how big the change is, the type of amino acid it is. Say for example, hemoglobin S means people having sickle cell anemia. Sickle cell hemoglobin is a change in the beta globin gene. As I told you, there is a change in the beta globin gene, where the actual codon at the sixth position is GAG. Okay, the actual codon at the sixth position in normal hemoglobin chain is GAG. Okay, because of change in the gene, the GAG codon is converted into GUG codon. Okay, as again I am telling you, if you look at the beta globin gene, in case of normal hemoglobin, person having normal hemoglobin, which we call as HbA, if you go from N terminal end towards the C terminal end, the sixth codon from N terminal end is GAG. Because of base pair substitution, what happens? GAG is converted into GUG. GAG codes for glutamic acid. So actual in the normal chain at the sixth position from N terminal end, there is glutamic acid. If GAG is converted into GUG, then GUG codes for valine. So the sixth amino acid, glutamic acid, is changed into valine, which is a hydrophobic amino acid. This amino acid is on the surface of the globin molecule exposed to water. Under low oxygen condition, valine's affinity for hydrophobic environment causes the hemoglobin to crystallize out of the solution. What happens when glutamic acid is replaced by valine, which is a hydrophobic amino acid, and under low oxygen tension, okay, the patient may be having various kind of problem, because in case of sickle cell anemia, the RBCs are sickle shaped and their lifespan is very less. They have low oxygen carrying capacity. Under low oxygen tension, they generally degenerate, right? So you can see in this diagram, RBCs of hemoglobin with normal hemoglobin person is round, and that of people having sickle cell anemia, it is crescent shape. Okay, crescent or sickle shaped we call it. Okay, if you look at the partial sequence, then you can see proline is at the fifth position from N terminal end, glutamic acid 6th and 7th. So at the sixth position, in case of normal hemoglobin, people having normal hemoglobin, it is glutamic acid. And if you look at people having sickle cell anemia, then at the sixth position, in case of people having sickle cell anemia, if you look at the sixth position, there is valine. There is valine at the sixth position, and it leads to sickle cell anemia. So people having sickle cell anemia suffer from various problems of oxygen transfer. the energy to work is less and all, right? So a small mutation, a missense mutation, changes a codon so that a different protein is created. Okay, and if a different protein is created having a single amino acid change, it can have a very drastic effect like that of sickle cell. So a single amino acid change can cause sickle cell anemia in a patient, and that is transmitted from generation to generation. So that can be a harmful effect of missense mutation, and which results because of base pair substitution, where there is only change in a single base pair. So this is missense mutation, right? And the other type of mutations are nonsense mutation, silent mutations, that we will study in the next lecture.